Welcome to this lecture on cell and molecular biology. Today, we are diving into one of the most crucial processes in all of life, the DNA replication machinery. Imagine trying to copy an entire encyclopedia by hand in just a few hours without making a single mistake. Your cells do exactly this every time they divide. We will explore the microscopic construction crew that makes this incredible feat possible, ensuring your genetic instruction manual is passed on flawlessly. To begin our journey, let us think about how this process actually works in nature. The DNA replication machinery, often called the replisome, operates exactly like a highly coordinated molecular assembly line. Why does this matter? Because copying billions of letters of genetic code cannot be done by a single worker. Instead, the cell hires a specialized team of microscopic workers, which are our enzymes and proteins. Each protein has one specific job, whether it is unzipping the DNA, holding it apart, copying the text, or gluing it all together. This division of labor is what allows your cells to duplicate their DNA with astonishing speed and flawless precision. Now that we understand the assembly line concept, let us look at the very first step of the process. Before we can copy the DNA, we have to open it up. This is where an enzyme called DNA helicase comes in, acting just like a molecular zipper. It breaks the hydrogen bonds between the two strands, creating a Y-shaped area known as the replication fork. But single-stranded DNA naturally wants to snap back together. To prevent this, single-strand binding proteins attach to the strands, keeping them safely apart. Meanwhile, unzipping creates intense twisting tension further down the line. An enzyme called topoisomerase, or DNA gyrase in bacteria, steps in ahead of the fork to relieve this tension, preventing the DNA from supercoiling and breaking. With the DNA successfully unzipped and stabilized, we can now begin the actual copying. The star of this show is an enzyme called DNA polymerase. Its job is to read the original template strand and add the correct complementary nucleotides to build the brand new strand. However, there is a catch. DNA polymerase is like a builder who can only add bricks to an existing wall. It absolutely cannot start from scratch. It needs a short starting block to get going. This is where another enzyme, called primase, comes to the rescue. Primase lays down a short sequence called an RNA primer. Once this primer is in place, DNA polymerase can attach to it and rapidly begin synthesizing the new DNA strand. As the new strands are being built, a unique problem arises on one side of the replication fork. Because of how DNA is structured, one of the new strands cannot be built continuously. Instead, it is constructed in short, fragmented chunks, which scientists call Okazaki fragments. If left like this, the DNA would fall apart, so a final worker is needed to finish the job. An enzyme called DNA ligase steps in, acting as a powerful molecular glue. It meticulously seals the gaps between all these Okazaki fragments. Thanks to DNA ligase, what started as a series of disconnected pieces is finally joined together, creating a perfectly continuous and unbroken DNA molecule, ready for the new cell. Before we move on to the mathematics of replication, let us see this entire team in action with a concrete visual. Here, you are looking at the replication fork. On the far right, notice topoisomerase relieving the tension ahead of the split. Right at the center of the Y shape is helicase, actively unzipping the double helix. Those small proteins clinging to the separated strands are the SSBPs, keeping the DNA open. Now, Look at the top strand, the leading strand, where DNA polymerase is smoothly and continuously building new DNA. Contrast this with the bottom strand, the lagging strand. Here you can clearly see primase laying down primers and the DNA being built in those short Okazaki fragments. Finally, on the far left of the lagging strand, DNA ligase is sealing the gaps to complete the process. Now that we have visualized how the replisome works, we must ask why understanding this machinery is so critical. DNA replication is the fundamental basis of all life, growth, and reproduction. Every time you scrape your knee, your body must build new skin cells, and each one requires a flawless copy of your DNA. If this machinery makes a mistake or breaks down, the consequences are severe, often leading to genetic diseases or cancer. But there is a silver lining. In the real world, doctors use this exact knowledge to save lives. Many chemotherapy agents and antibiotics are designed to specifically target and jam the DNA replication machinery in rapidly dividing tumor cells or bacteria, effectively stopping the disease from spreading. This understanding of the machinery's speed leads us to an important mathematical relationship. How long does it actually take to copy an entire genome? We can calculate this using a simple formula. T equals N divided by the product of R and M. Conceptually, the time it takes depends on how big the genome is, divided by how fast the enzymes work, and how many teams are working simultaneously. Here, T is the total replication time in seconds. N is the total number of base pairs that need to be copied. R is the replication rate, which is the speed of a single DNA polymerase enzyme in base pairs per second. Finally, M is the number of active replication forks. If the genome size N increases, the time goes up. But if the cell uses more forks, M, or a faster polymerase, R, the total time will decrease. Now that we have our formula, let us see this with a concrete example, using a real bacterial genome. We are given an E. coli bacterium with a single circular chromosome containing 4.6 million base pairs. We are also told its DNA polymerase operates at a rapid rate of 1,000 base pairs per second. And because the chromosome is circular, it uses 2 replication forks. We need to find the total time to replicate the genome. We chose our time formula because it perfectly links genome size, speed, and fork number. First, we substitute our given values into the equation. Next, we multiply the rate by the number of forks, showing the combined machinery processes 2,000 base pairs every second. Dividing the total base pairs by this combined rate gives us 2,300 seconds. Finally, dividing by 60 gives us our final answer of approximately 38.3 minutes. This physically means an E. coli cell can completely duplicate its entire genetic code in under 40 minutes. Having calculated the overall time, let us zoom back in on the lagging strand, which brings us to another important calculation. Remember that the lagging strand is synthesized discontinuously. We can estimate exactly how many of these pieces are made using the formula F equals N lagging divided by L fragment. Conceptually, if you know the total length of the track and the length of each stepping stone, you can find the number of stones. Here, F represents the total number of Okazaki fragments. N lagging is the total number of base pairs on the lagging strand, which is equal to the entire length of the chromosome since it spans the whole DNA molecule. L fragment is the length of one individual Okazaki fragment. If the fragment length decreases, the total number of fragments the cell must manage will drastically increase. Before we conclude, let us apply this fragment formula to a human chromosome. We are given that a single human chromosome contains 150 million base pairs, and the average length of a eukaryotic Okazaki fragment is 150 base pairs. We are asked to find how many fragments must be synthesized and ligated. We use our fragment formula because it directly relates total strand length to individual fragment size. We substitute 150 million for the total base pairs on the lagging strand and 150 for the length of each fragment. Dividing these numbers yields exactly 1 million fragments. Physically, this final answer means that for just one single chromosome, the DNA ligase enzyme must perform its gluing action 1 million times. This perfectly highlights why the replication machinery must be both incredibly fast and astonishingly precise. We have covered a lot of ground today, so let us summarize the most important ideas from this topic. First, always remember that DNA replication is not a random process. It is a highly coordinated molecular assembly line. Second, this incredible feat is made possible by dividing the labor among specialized enzymes, with helicase unzipping, polymerase copying, and ligase sealing the DNA. Third, we saw through our calculations just how fast and precise this machinery must be to copy millions of base pairs in minutes. Finally, this knowledge is not just theoretical. It provides crucial targets for treating severe diseases like cancer and bacterial infections. You have now mastered the fundamentals of the DNA replication machinery. Great job, and keep exploring the wonders of molecular biology.